So Marlene, thank you so much for the introduction. Hudson, thanks for the invitation. It's really fantastic to be back in La Jolla and at Sanford-Burnham. And what I want to talk today about is protein quality control and aging. And I also promise to talk about some sugars as I go. So just as a broad introduction, why would we study the biology of aging in the first place? And it is not only that we lose the positive attributes of youth as we age, as we become older, but also aging is really a major risk factor for diseases. And what's important is that aging predisposes for very distinct diseases. And here I'm just showing the cancer rates and dementia rates in Europe. And you can clearly see, and I guess all of us know that really, that these diseases mostly occur in the elderly population. And so we study aging to really understand the underlying risk factors for age-related diseases, to really get at the reasons why these diseases might actually occur, what lets them actually occur, and why in old people. And so specifically, as we get older, mechanisms actually begin to lose their function. So in young organisms, as you all know, regeneration capacity, stem cell function is very good, so there's a high fidelity of molecular quality control. But as we get older, these lose their function, and this leads to aging and age-related disease. So among the molecular quality control mechanisms, one particular one that I'm focusing is protein homeostasis, or protein quality control. And now, protein quality control describes the combined mechanisms that generate and maintain a healthy proteome. And this includes protein biogenesis, protein modifications, mostly post-translational modifications, such as glycosylation, but also the management of protein aggregates and the degradation of toxic proteins and aggregated proteins. And now, protein quality has to be a very well-balanced process, and cells, and this occurs in diseases such as Huntington's or Alzheimer's disease. And so it has previously been shown that in long-lived mutants, in very many laboratory species, protein quality control is actually something that's activated. So there was always this correlation between the two. And what we wanted to find out in this project was a very simple question. This is whether there's also a causative link, whether we can actually improve protein quality control, improve protein homeostasis, and ask whether that is causative for lifespan extension. And so we addressed that question in the very simple model organism C. elegans. It's a very small worm that typically lives in the soil. It's just about 1 millimeter in size, actually. But it's really a fantastic organism for aging research. First, because due to the small size, we can keep lots of them. They actually reproduce very rapidly, and one adult animal actually gives rise to about 250 offspring, so we can expand them quickly. The genetics are very well understood. The genome is sequenced, and we have very nice genetic methods to work with the worm. Okay, so to address protein homeostasis, then we turn to one particular organelle, and that's the endoplasmic reticulum. And we did that because all the proteins that are secreted from cells and all cell surface proteins are actually synthesized and processed in the ER. So the ER is really a major site for protein quality control and to challenges to protein homeostasis. And this slide, and I want to point out, I actually designed this slide also myself, just to keep with the topic of the day, just to get you up to speed to what occurs during protein folding in the ER. So actually, proteins are imported into the ER lumen co-translationally, and then these nascent peptides depend on a number of activities, including the activity of chaperones, disulfide bonds, of course, energy, to reach a fully folded state. And one very important modification in the ER, and a specific ER modification, is the addition of N-glycans or N-glycosylation. Well, fully folded proteins are then shipped off to their target destinations via the Golgi apparatus, but of course protein folding is a messy business, so folded proteins are actually in some sort of an equilibrium with misfolded proteins. And so misfolded proteins have to be refolded, but sometimes this terminal misfolding of proteins then makes it necessary for them to be degraded. And these unfolded proteins then activate the unfolded protein response, which is a signaling cascade that signals back to the nucleus. toxic proteins are then actually not degraded inside the ER, but they're actually retrotranslocated back into the cytoplasm in a process called ER-associated degradation, and in the cytoplasm then they're degraded by the proteasome. So we wanted to use the worm to find ways to improve protein quality control. And so to do that, we used a pretty simple trick. So we posed a certain challenge to these animals, and that challenge is a drug called tunicamycin. Tunicamycin powerfully inhibits N-glycosylation and is therefore a major trigger for protein quality control stress, essentially, in the ER. And it's toxic for worm development. So we simply asked whether we can generate mutants that can survive the toxic environment in the presence of tunicamycin. So we did a classic forward genetic mutagenesis screen in which we introduced point mutations randomly across the worm genome, so we can really query the whole genetic space in the animal. And then one generation removed, we performed a developmental tunicamycin selection screen. And the benefit of the screen was really a very simple readout because we had really an all or nothing effect. So wild-type animals or non-resistant mutants will never really develop under these conditions, whereas drug-resistant mutants can not only develop, but they're actually also fertile. So we can actually even maintain them in the high tunicamycin levels. So a very strong phenotype here. And so the drug-resistant mutants we then identified, we used next-generation sequencing to identify the causative mutations. And one gene that caught our attention early was glutamine fructose 6-phosphate aminotransferase, or GFAT1. And the interesting thing was that we actually identified independent alleles of GFAT1 in our screen. So independent tunicamycin resistance strains actually turned out to have GFAT mutations. And these are all labeled here. So we looked at 3 independent mutations, and all of these are actually single amino acid substitutions. GFAT1, as many of you guys fortunately know, typically I speak to an audience where I actually have to explain what the hexosamine pathway is. So it's really nice. So the hexosamine pathway is really regulated by GFAT1. It is the key enzyme and the only regulated enzyme in the hexosamine pathway. And GFAT1 takes fructose 6-phosphate from glycolysis and builds up UDP N-acetylglucosamine, or UDP GlcNAc. And this was really kind of exciting, or interesting at least, because this is exactly where tunicamycin also acts. So it seems like in our screen we actually hit a gene that was very close to the effect that we had with the drug. And to really make sense of this, we hypothesized that the mutations we had in GFAT are gain-of-function mutations. So this would explain the observation that we have tunicamycin resistance. We hypothesized then that GFAT would be more active, that would increase UDP GlcNAc levels, and that would simply counter or neutralize tunicamycin and therefore counter tunicamycin. For example, this was conserved genetically. We found that these gain-of-function mutations are actually genetically dominant. But more interestingly, I think we were actually able to measure UDP GlcNAc using LCMS. And so you can see here in color that all 3 mutants actually had elevated UDP GlcNAc levels compared to the wild type in black. And this is quantified over here, both in young larval animals as well as in the adult. All 3 GFAT mutations were gain-of-function. So we actually then went on to characterize that at the level of N-glycosylation. And to do that, we used a lectin. This is a concavalin A, which is a lectin that binds specifically to N-glycans. So we can simply run a Western blot and using this lectin fused to HRP, look at the N-glycosylated proteins. And consistent with our expectation that tunicamycin inhibits N-glycosylation, we see that in the wild type there's a significantly reduced signal, but in GFAT gain-of-function mutants, they're completely resistant. So they're not only resistant regarding their development, but also at the level of N-glycosylation, we can again see that resistance. So in the presence of tunicamycin, all of these guys really have no difference in N-glycosylation levels, and this is quantified over here. So in all, we were like, well, this is sort of interesting but predictable. So we sort of got what we screened for. So we have a gain-of-function mutation in GFAT, and this counters tunicamycin resistance down here. So we were wondering if we should follow this, but then the story got exciting when we realized that in the absence of tunicamycin, just under normal conditions, the GFAT gain-of-function animals were actually long-lived. So these are the survival demographics that we do with worms. So we take a whole population of animals, and then we just really follow their survival. And with these survival curves, we then can compare wild types with GFAT gain-of-function animals and see that they're actually long-lived. And this is true for all three mutations. And this is in the absence of tunicamycin, so just under normal control laboratory conditions. So we then went on to really ask whether this is due to the activity of GFAT1 in the hexosamine pathway. So we also overexpress GFAT1, for example, in a transgenic worm. So we see lifespan extension simply by elevating copy number of GFAT. And most excitingly to me, we can actually extend lifespan by feeding the precursor of UDP GlcNAc in the media to worms. So we can feed GlcNAc, and that gets us the lifespan extension as well. Here I have to say that we actually had to use pretty high levels of GlcNAc to get that effect, but we used a osmolarity control, the arginine, which had basically a control lifespan. So you can see that really the activation of the hexosamine pathway, either genetically or in this case by supplementation, extends lifespan of the worm. We wanted to then look at that in more detail at the level of protein folding in the ER to really get at the mechanism. And therefore, we turn to a very, I think it's a really elegant model to actually look at protein quality control in live worms in the ER. So it's really a protein folding sensor. This model uses the neuroserpin SRP2, which is here expressed as the point mutant, which leads to misfolding of this protein. So this protein is slightly aggregation prone. And then this protein is fused to GFP, to YFP, to make it visible and driven specifically in the muscle into the worm. So here you see the head region of a worm, and we can actually observe these single aggregates within the lumen of the ER in a live worm without any staining. So we can really watch protein folding in the ER with this model. And when we crossed this transgene then into the GFAT gain-of-function background, we saw that the number of these aggregates was significantly reduced. And this was also significant after statistical analysis, actually across a pretty long time during the worm's life. And we could also get this effect by feeding GlcNAc, interestingly. So we were then asking what the mechanisms might be. What are the mechanisms that degrade aggregated proteins in the ER? And so we tested these systematically. So it's known that aggregates in the ER are degraded by autophagy via ERAD, as I presented previously, and they activate the unfolded protein response. So we're wondering what is the mechanism that then makes these worms long-lived. And to really summarize 2 years of work, we found that the unfolded protein response was not changed, but we found elevated levels of components of the retrotranslocan in ERAD and also elevated proteasome activity. And we also found activated autophagy. So it seems that we really have a change in proteolysis, in proteolytic processes in these worms that makes them long-lived. And we were wondering then if these processes actually are required for the longevity that we see. And so we used RNA interference in the worm, and we knocked out a component of the ERAD machinery. So in black is the wild-type lifespan, in blue the extended GFAT gain-of-function lifespan. And then when we knock out an ERAD component, we reduce the lifespan back to wild-type. And we found a similar phenomenon when we knocked out or knocked down autophagy using ATG18 RNAi, which inhibits autophagy. So again, the extended lifespan in the GFAT gain-of-function here in green is reduced to wild-type levels. And this is kind of interesting because it's not a partial reduction, but whenever we knock out either of these processes, we lose the complete lifespan extension. So we're still trying to understand how that works. So next, we asked, well, autophagy and the proteasome are not really exclusive in the service of the ER, but really they degrade toxic proteins or toxic proteins also from other compartments. So we were wondering if we also had a benefit in compartments outside the ER. So we looked at a toxic protein model in the cytoplasm. And for that we turned to another transgenic worm, which expresses a polyglutamine peptide in the muscle. So polyglutamine peptides tend to aggregate because they're inherently sticky, and they're also involved in the etiology of Huntington's disease. And here this protein was expressed in the muscle, so we can look at proteotoxicity really and use the readout of motility of the worm to observe that. And so we can see that we saw that when we actually feed these worms with the arginine at an age of 10 days, they're still alive. See, it still moves, but not very much. And then when we actually feed GlcNAc throughout life for this 10 days, motility is pretty much is maintained. And so we also can quantify that statistically in motility assays and see that GlcNAc or UDP GlcNAc treatment actually significantly increases motility. And this was also true in the GFAT gain-of-function mutants, so also in the genetic model. So to sum this up, what we found is that we can activate the hexosamine pathway in a number of ways. One, genetically, by GFAT gain-of-function, or by actually feeding UDP GlcNAc precursor GlcNAc in the diet. This leads to elevated UDP GlcNAc levels, and really through a process that we haven't understood yet, we get a reprogramming, if you will, of protein quality control. So we get elevated autophagy, elevated ERAD, and elevated proteasome activity. And together, it seems these processes are then required to counter toxic protein aggregation and to extend lifespan. So after we sort of finished this story, I was fortunate enough to start my own lab at our institute, and this is just about 5 months ago. So I don't have a lot of new data to share with you since we started, but I just want to give you like a few ideas about how we're addressing this and how we want to move this project into the mammalian system. And one of them, just as an introduction, is that GFAT is highly conserved. It's pretty amazing. So here's the peptide sequence surrounding the gain-of-function positions in GFAT across basically the whole zoo of my most favorite animals. And down here, I put in the C. elegans sequence the respective point mutations of these alleles. And now, not only in the direct vicinity of the gain-of-function mutations, we have this really high conservation, but all over GFAT is about 80% similar at the amino acid level between the worm and us. So I'm hoping that not only the functional properties of GFAT in the pathway, but also its physiology is conserved. And so we're testing that in the mammalian system now. And one thing I did was a proteasome activity assay, so we can actually measure proteasome activity by looking at the turnover of a fluorescent substrate as a function of time. And what I observed was a dose-dependent increase in proteasome activity as we incubated these cells with GlcNAc. So it seems that we can actually activate the hexosamine pathway in cells, and that leads, as it does in the worm, to elevated proteasome activity. And another thing, and this is really just a tool that we're developing, but I'm really excited about it, so I have to tell you, is that we can actually overexpress GFAT1 in cultured cells, and that elevates UDP GlcNAc. So it's again very similar to the system in the nematodes, but more excitingly, we can use the CRISPR-Cas9 system to actually do genome editing. And what we actually did here was to introduce exactly the point mutations that activate GFAT in the worm in the homologous positions in the mouse genome in these neuronal cells. So really, we have these single amino acid point mutations we put into the mouse genome, that and only that. So it's a very elegant way to actually activate the locus in these cells. And so what we see when we put in this mutation in two independent lines is a significant elevation of UDP GlcNAc levels. And so as we speak, these experiments are going on to actually now ask if in the background of these cells we have improved proteome quality control, if we get reduced toxicity and all these things. And I hope in a few months I could be able to tell you more about this. So with this, I'm coming to an end. I really want to acknowledge my postdoc mentor, Adam Antebi, as well as Nadia Storm, who was working with me on this project during my postdoc. And I want to point out also that Nadia is actually also an ex-student in Malene's lab and at Sanford-Burnham. So it's all coming full circle here. But more so, I actually want to acknowledge the people in my lab who took the risk to join a brand new PI. And this is the team as we are currently in the lab. And this is the team 3 weeks ago during the Cologne Carnival. So with this, I would like to, again, thank you so much for inviting me, and it's a real pleasure, and I'll be ready to take any questions. from DeepT was whether there's a tissue-specific effect of GFAD1 activation. So whether essentially we can overexpress, for example, GFAD1 in one tissue and see whether there's cell non-autonomous effects to extend lifespan. Pretty much what you meant, right? So that's an experiment that's going on in Adam's lab currently. So they're overexpressing GFAD1 systematically to see if certain tissues are required or whether there's actually cell non-autonomous effects. Yeah. That's very important, and we haven't done it as a function of age. We looked at that at one certain time point simply to ascertain whether we don't have changes in the expression levels of the transgenes and whether that would be responsible for the effects. So we didn't have any changes in expression, but actually we couldn't detect the great difference between aggregated SDS insoluble material versus soluble material at that point. So that biochemistry is something I do want to follow up in the cell system now, because in the whole worm it's of course more complex.
I was wondering, from an evolutionary perspective, why those are so conserved. Is there any downside of this gain of function mutation that you have, or whatever that is? Why didn't evolution create this gain of function mutation? Did you find it? Everything showed that it makes the worms better, and
right, and yeah, we actually looked. So many longevity models do show some sort of a trade-off, right? So many long-lived worm mutants actually have lower capacity to lay eggs or whatever. So we were looking whether there's such a trade-off anywhere, but we didn't see that. So they had really similar phenotypes as wild types besides the longevity. The only answer I have, and that's pure speculation, is that wouldn't be selected for during the reproductive period. And so it's a benefit that only comes out late in life. I'm actually in touch with some geneticists who have human sequences to see if in long-lived humans there might be SNPs in GFAD that might activate it, but I don't have anything on that yet.
So we know that tunicamycin causes the absence of glycosylation site occupancy. So I was wondering if you did any kind of proteomic analysis to look at site occupancy. You know, when you give this, I mean, are more sites occupied, something like this?
The answer is no. We didn't do that yet, but it's definitely one of the hypotheses we're testing. So in my model, we left the most important question, right? So we have elevated UDP GlcNAc, and then we have all these beneficial degradative processes, and we really don't know what happens in that black box in the middle. And one explanation would be, of course, that we improve protein folding or some sort of a signaling effect by site occupancy, for example. But other hypotheses would be then, of course, branching, O-glycosylation, N-glycosylation. We really have to look at that now. And so some experiments are going on in the worm, but I think, again, the cellular model is going to be cleaner and very useful for that. And we're getting that started right now.
Do you have loss of function in that? Does it
Can you say again, loss of function?
Yeah. So basically, so giving low calorie, caloric restriction. That one efficiency does it counteract that?
So we did one experiment where we wanted to address whether the hexosamine pathway would be a pathway that other longevity pathways sort of signal through. So we measured by LCMS the UDP GlcNAc levels in pretty much all the long-lived strains that we could get our hands on in the lab, and none of them had elevated levels. actually only the E2 worm, which is the model for dietary restriction, had a little bit of a lower level of UDP GlcNAc consistent with lower food intake. So from that, we think that known longevity pathways don't go through the hexosamine pathway.
Any more questions? That would be a good transition to lunch, I guess, from dietary restriction. So lunch, first of all, let's thank all the speakers for a great talk this morning. And I believe lunch will be served right outside.
And then the table's out of the freezing cold where you can eat.
Yes.
There is a little lounge right up above the entryway that has a number.
So enjoy your lunch and please be back at 1:30.
Freezing cold in San Diego.
Very good. Yeah, I know. It's